Hello everybody, it's Mrs. Pound and we are studying Chapter 7 on Biotechnology from BJU Biology's 4th edition. We have been taking a look at Section 7b on practical uses of biotechnology. And we're going to talk today about subsection 7.5 on gene therapy. So our objectives today will be to define gene therapy and discuss how gene therapy could be used in humans. Gene therapy is a technique to correct a defective gene that has been identified as the cause of a specific disease. So its goal is to replace a malfunctioning gene with one that functions properly, kind of like changing a light bulb that has burned out. If we have a light bulb that doesn't work, we switch it. So gene therapy is an attempt to do the same thing. Now, there are a couple of ways that this can be done. There can be cells that are grown in the lab, and then the new cells are injected into the individual, perhaps stem cells from the stem cell technology that we talked about before, that if an individual, say their liver was not functioning properly, stem cells that differentiate into a liver could be grown and then placed into the individual, basically kind of transplanted into them. The other way is to introduce a viral vector that injects DNA fragments into the cells and replaces the fragments that it has with the defective fragments in the individual's cells. There are many potential problems with gene therapy. The first is it seems to have a short duration. The few gene therapies that have been attempted usually have to be repeated because the effect, the desired effect, is lost after a couple of cell divisions. So it's not very long lasting. The second problem is that the viral vectors may lead to infection. We try to take out the infection part of the virus, but something might be missed, and so then instead of just it being a good thing and treating the genetic disorder, the individual could be made sick by giving them a virus. The body recognizes the viral vector as a foreign substance and destroying it. So viruses, part of our immune system's job is to identify viruses and destroy them. Now, the first time the virus is introduced, it may be just fine, but it's the second time when the immune system recognizes it faster that there could be an issue, and so then it does nothing. And some diseases are caused by multiple gene interactions. So with that, even if you replace one gene, there might be another gene that also has to do with the disease. So there may be more than one gene that needs to be replaced, and it could be a much more complicated issue than we realize. So our objectives today were to define gene therapy, where we replace a defective gene with a good gene, and discuss how gene therapy could be used in humans. It could be used to cure genetic disorders. It could be used to cure cancer, but there are many issues that we have to work out. It's a little more complex than it seems. So don't forget your 5 questions in the margin and keep watching to answer the questions at the end.